Okay, before we start this video, I want to give a quick disclaimer. This video's main purpose is to demonstrate how to perform a blood count using a Leishman stain, and then to see whether or not I had more white blood cells present after my COVID-19 vaccine. It's not to show you that if you perform this experiment at home after your vaccine and you get a different result, that your vaccine didn't work or something is wrong with you. The symptoms associated with the COVID-19 vaccines vary greatly from no symptoms at all to a small fever and some aches like I had to full-blown flu-like symptoms. However, this is a small price to pay to stop a virus that managed to straight up end life as we know it. So TLDR, we're looking at my blood today and go get your vaccine. Also, if you get a bit squeamish looking at blood coming out of people, I'll make sure to put a chapter to warn you when the blood is coming up, so just skip that. With all that out of the way, welcome guys, gals, and non-binary pals to this month's episode of Scuffed Science, where today we will be looking at how to stain a blood sample and whether or not I had an elevated white blood cell count after the COVID-19 vaccine. I love hematology and immunology, so much so that this fall I'm going to college for a bachelor's degree in medical lab sciences with the goal of eventually becoming a physician's assistant in one of these specialties. Hematology is so interesting to me because blood is deceptively complicated. I think most of us know that blood consists of things like plasma and platelets, white blood cells, and the star of the show, red blood cells. But I didn't originally know that there are different types of white blood cells with different functions. This image right here contains most of the different types of blood cells and where in the body that they come from. Okay, let's break this down real quick. So we start off with blood stem cells, which then turn into either myeloid or lymphoid stem cells. Myeloid is a Greek word that literally means resembling or coming from bone marrow. Bone marrow is the squishy material located inside your bones where almost all of your blood cells come from. I say almost because of the other type of blood cells. Lymphoid cells have one specific cell type that just had to be different. Lymphoid cells differentiate into two different cell types, T lymphocytes and B lymphocytes. By the way, these cells help to form your lymph system, whose function is basically like those detox teas celebrities promote, just without the bowel movements from hell. T lymphocytes are a cell type that is different from the rest. Technically, they do start their existence in bone marrow, but much like your trust fund baby friend, they inevitably go on an expensive vacation using daddy's credit card, but instead of riding elephants in Thailand, it's becoming immunocompetent in the thymus. Because T lymphocytes are the only blood cells to become immunocompetent outside of the bone marrow, then the B and B lymphocytes must mean bone marrow, right? That would make the most sense, but actually the B stands for bursa and refers to the fact that these cells were discovered in the bursa of some birds by a couple of nerd ornithologists. Myeloid cells have many more different types of cells, including platelets or thrombocytes if you want to be scientific, that help to stop bleeding and clot blood. Red blood cells are erythrocytes that carry things like oxygen and nutrients through the body, and the cells I'm most focused on today, leukocytes, meaning white blood cells. Leukocytes have four main types of cells that I want to teach you about in this video. Macrophages are cells whose main function is to eat bacteria and other pathogenic organisms. Honestly, they look and function a lot like amoebas, sliding around and hunting and eating bacteria. Eosinophils, basophils, and neutrophils are the other white blood cells important to your body's immune response. Eosinophils manage responses to parasitic infections. Neutrophils are similar to macrophages in that they also eat bacteria before dying in a blaze of glory, in an effort to make sure those pathogens don't continue reproducing. And basophils, who release histamine when presented with an allergen, causing that oh-so-familiar runny nose and tickly throat, which is quickly quelled with some Benadryl. Benadryl, because you can't have allergies while you're unconscious. Something I also want to talk to you about is those little dots you see inside the cells. Those are called granules. Now, not every white blood cell has these, however they are extremely important to the functions of these neutrophils, basophils, and eosinophils. Granules contain antimicrobial agents that kill things like bacteria and viruses and help in the process of phagocytosis. Man, it's kinda crazy how these cells devote their entire existence to protecting me. They exist only for a few fleeting moments on this blue marble, a small speck in the vast and infinite universe, existing for mere months, sometimes weeks, molded by a constant war against pathogens attempting to kill me, saving me anytime I roll the dice on crappy gas station pizza, or the day-old milk I forgot to put in the fridge. They sacrifice everything for me, to keep me safe, to keep me living. What a bunch of idiots! I won't dive too much into how exactly the vaccine works when inside your body, as I plan on doing that in a different video, but I do want to focus on how our immune systems respond when presented with a foreign object. For an immune response to occur, there needs to be an antigen that alerts your immune system that something is up. Your immune system is divided into two main groups with different functions. The myeloid cells all fall under your innate immune system. These cells are the first responders of your immune system. They rush to the site of infection and start the process of phagocytosis. Most of the time, that's it. The cells kill off the small infection and then teach your lymphocytes about the infection so it doesn't happen again. However, sometimes the task is too tall for the innate immune system to handle alone. Macrophages hit the panic button, causing the hypothalamus to increase your body temperature. Basophils and specialized mast cells start to release histamine, which dilates your blood vessels and causes inflammation. Wait a minute, aches caused by inflammation? Fever? These are the same symptoms I get whenever I get a cold. Yes, my dear friends, the symptoms you feel from the common cold or the second dose of the vaccine aren't caused by the pathogen itself, but by your immune system using things like chemokines to communicate and help guide white blood cells to fight infection. I did say that there were two components to your immune system, and that second portion is called your adaptive immune system. This type of immunity is a pure masterclass of genius by your body. To heck with the Mona Lisa, put this in the Louvre. The adaptive immunity is composed of your lymphocytes. Whenever you hear about antibodies when referring to COVID-19, they actually come from the lymphocytes of the adaptive immune system. The innate immune system's phagocytes present naive lymphocytes with specific markers of the pathogens that they've eaten. B and T lymphocytes then start producing specific protein complexes called antibodies, with other cells having the specific job at remembering the pathogen and how to stop it. So when the body sees the same pathogen in the future, lymphocytes can come to the rescue and release antibodies who one-punch man any invaders. Understanding how these different immune systems respond is extremely important to my theory for how my blood will look after the second shot of the vaccine. I expect to see an increase in free-floating leukocytes as my immune system responds to the COVID spike protein. However, I'm also interested in seeing how many lymphocytes are present. After all, my body has spent the last couple of weeks teaching my naive lymphocytes about what the virus looks like, so maybe some lymphocytes are already releasing COVID antibodies. Phew, that's all of the nerd stuff out of the way. Now I understand why Michael Rees leaves out all the coding elements whenever he does a video. Anyway, on to the actual project. Today we are going to use Leishman stain to stain the nuclei of white blood cells. Okay, just like a little bit more nerd stuff. Leishman stain belong to a group of stains called Romanowsky stains. All of these stains are very important for identifying blood abnormalities and diseases. I've probably shown you some pictures of proper Leishman stained blood throughout this video, and you'll notice that the nuclei of the cells are blue. This is because the stain is composed of eosin and methylene blue, which is very important. Both of these chemicals will be featured heavily on the channel in future projects. Eosin is acidic, while methylene blue is basic. Speaking of basic, I'm assuming most of you have a baseline knowledge about the pH scale. And if you don't, you should tune in next month where I'll go into more depth about it. The nuclei are stained blue because they are basophilic and attract the methylene blue. I believe nuclei are basophilic because they are acids that attract and bind the opposite side of the pH scale. Think about it. Nuclei manage DNA. DNA. Deoxyribonucleic acid. Pretty cool, right? Okay, this time we'll go into the procedures for real. First, you'll need some important materials. Obviously, you'll need some Leishman stain, a sterile lancet to draw blood, some sterile alcohol wipes or rubbing alcohol, a couple of glass microscope slides, a cup or container for your stain, and a microscope. Most Leishman stains that are purchasable online come powderized, and so it needs to be rehydrated. This is where I messed up. I got the protocols for the stain mixed up and used demineralized water instead of methanol. This didn't end up affecting the stain too much. The overall color was a bit different and more muted. However, I ended up adding some eosin Y I had laying around, which worked pretty well. The slide I stained before my shot when I had just added the eosin was a lot more pink than the second shot, which appeared more of a blue color at 40x magnification. Don't fret though, this has nothing to do with the vaccine and everything to do with me not being patient and letting the eosin settle into the solution like it did when I used the same solution for the second shot. To actually get the blood on the slide, first you want to make sure that your workspace is sterile. I did this by wiping down my arms and work area with isopropyl alcohol and getting 2 fresh out of the box glass slides. I then put some more isopropyl on the finger I would be taking the sample from. Before this, I loaded my lancet with a new needle. I jacked the spring up to 4 to make sure I got some good blood flow and promptly exsanguinated myself in the name of science. Our work isn't even close to being done though. I squeezed out a drop of blood and placed it on the right hand side of my slide. I took the second glass slide and performed a not very good thin blood smear. That's a yikes for me, dog. Make sure you do better. After letting the blood dry for a minute, I applied the stain with a sterile pipette. I've used both cups and petri dishes for this portion and both work pretty well. After 10 more minutes, we wash off the excess stain and let it air dry. That's pretty much it, honestly. A pretty simple stain that takes about 20 minutes to complete. Alright, this is what everyone is here for. Was there actually any difference? Well, yes, actually. And the results that I got were super interesting. Here are the 2 slides side by side. At the lowest magnification, I didn't see much of a difference. Both slides have a lot of blackish dots. Most of these are actually the large dark stains of the lymphocytes and other white blood cells. However, at higher magnifications, there were a lot more visible white blood cells after my shot, when I was feeling the aches and had the fever. So yeah, the slide with my blood after the second shot had a lot more white blood cells, including lymphocytes. Just in case you didn't believe me, I bought a microscope to phone adapter to get some higher quality video. Here, look at these 2 photos taken on the same magnification. The slide before the shot with the visible eosin had really visible red blood cells, but not nearly as many visible white blood cells. I highlighted all the white blood cells I could see in Illustrator and put the number at the bottom. If I was to actually do a real experiment trying to find out what happens to blood after a vaccine, I'd get a randomized sample of about 100 people from all different demographics. I'd also take a bit more blood than the singular drop I used for my stain. I'd use one of those fancy cell counters that would give me quick empirical evidence if there was any major changes across a group of people. But beggars can't be choosers, and if I said it once, I've said it 1,000 times. This is Scuffed science, baby. My home lab is my room where my mom brings me chicken nuggies. The production budget for this video was like a crisp $10 bill. That's it for me. In today's video, we explored a variety of topics. This includes what makes up your blood, what your immune system is and how it reacts when presented with a pathogen, as well as how to perform a Leishman stain. Make sure you smash that dislike button and unsubscribe from the channel because this video was probably garbage. And remember to call your mom. I'm recording this on Mother's Day, so think about the good things she's done and tell her I say hi.